last type of cell surface receptor is enzyme-linked receptor. So this means that these cell surface receptors are linked to enzymes. Okay, so here's an example. This is a receptor tyrosine kinase. So tyrosine is a type of amino acid, and a kinase, based on the name, with ASE, you know that it's probably an enzyme, right? You remember those enzyme names usually end up with ASE. Okay, so here's how it works. Now these tyrosine kinase receptors kind of span through the cell membrane, right? So that's why they're cell surface receptors. And these red dots are going to be the ligand, okay, the signaling molecule. So these ligands are going to bind to the receptor, all right? Now in the receptor, there are this enzyme component. So this is the enzyme component for this receptor, and then this is the other receptor, all right? So when the binding takes place, the receptors are going to dimerize, okay? And they are going to activate the enzyme portion, this portion right here. So the enzymes will be phosphorylated over here, okay? Phosphorylated, meaning a phosphate group is added to the receptor. Okay. Phosphorylated phosphate group added to the enzyme. So the enzyme is activated once the phosphate group is added. All right. Now the enzyme portion of the receptors can activate other proteins. And then these proteins will eventually trigger a cellular response. So again, this is the ultimate outcome, and it's going to be, through a series of pathways. But first of all, you have to activate the enzyme portion of the receptor, and then that enzyme will activate other proteins, other enzymes, and eventually that's going to lead to a cellular response. Okay, so this is how enzyme-linked receptors work. All right, this is an example about the G protein coupled receptors. Really, it's about G proteins, not necessarily the receptors. So just real quick, you guys probably have heard of the disease called cholera, right? So caused by a type of bacteria. So the bacteria makes you sick by producing a toxin. So we're just going to call it cholera toxin. Okay? People infected with this bacteria are going to have a series of gastrointestinal symptoms. So diarrhea is very common. And this is how the toxin can cause diarrhea. Okay, so the toxin has two parts. Okay, so here it's shown, this is the structure of the toxin. So the toxin is a protein. Okay, so one of the sections of the protein, section A, can go through these receptors and then enter the cell. Okay, so that's the A. Okay. And then it's going to activate the G protein right here. So activate the G protein. Okay, and then activate. Can't spell. Now, if you remember, we said the G protein, once activated, the subunits will dissociate, right? And then one or both of them can activate other enzymes, which will eventually cause a cellular response. Okay, so the toxin can activate the G protein, and then the G protein activates this enzyme, okay, which activates a second messenger. So this is the first messenger first, and then this is the second messenger. And then this second messenger is going to lead to a cascade of a response. But eventually, the cellular response is going to be the continuous opening of chloride ions, and probably some other ions as well. But the main one is the chloride. So I'm just going to Cl chloride ion gates. Okay. It's really a protein channel. So you can say chloride ion channel. All right. So This series of pathways eventually is going to cause the continuous opening of the chloride ion channel. So if you remember from the gated ion channel receptor, if the channel is open, then the ion is going to diffuse, right, following the concentration gradient. So you have a lot of chloride, okay, inside the cells lining your intestines. Okay, so high concentration inside and low concentration outside the cell. So chloride is going to diffuse, it's going to flow out of the lining cells. And then water is going to follow chloride. Remember the osmosis section, we talk about water wants to go from low solute to high solute, right, to kind of bring down the concentration. So as the chloride leaves the intestinal cells, so water is going to follow, and this is going to cause too much fluid in the feces. So this is what diarrhea is, right? Too much water. So a lot of patients infected with cholera will have severe dehydration. So one way to kind of help them is to supply them with a fluid so that they are not severely dehydrated. Okay. All right. So this slide looks kind of a little bit busy, right? Especially this figure right here. So the message, really the take-home message from this slide is that the initial binding, okay, especially for the cell surface receptors, the binding is going to initiate a signaling pathway, right? So the signaling pathway can be very complicated. There are going to be, so many proteins that are activated or deactivated, and eventually gene expression is probably going to be altered. Okay, so let me just use this figure as an example. Okay, so I know there are a lot of different proteins in this figure, so just kind of follow me. What you have is going to be EGF, so this is going to be the signaling molecule, the ligand. Okay, so EGF stands for the epidermal growth factor. So this EGF is going to kind of really promote cell division. Okay, so it's a growth factor, right? So if the cell receives this signal, the cell is going to think, okay, I need to divide faster, okay, make more cells. All right, so how does it make cells divide faster? Okay, so it's going to bind to the cell surface receptor, okay, and then the enzyme portion of the receptor is activated right here, and then this enzyme is going to activate other enzymes. Okay, so it's going to activate GRB2 and SOS. And then these enzymes, okay, so that's one, right? And then the second step, these enzymes are going to activate another protein, the RUS, right here, the purple one. So that's a small G protein. And then once the RUS is activated, it's going to, how do you know it's activated? See, it's linked to GTP, right? Not GDP, so it's activated. Now this activated RUS protein is going to activate another protein, another enzyme RAF right here. And this enzyme is going to phosphorylate or activate. So the same thing, activate. Another group of enzymes, right? So they're over here. So that's the MEK. And then the MEK is going to activate another enzyme. And then eventually these things will get into the nucleus and then affect gene expression. Okay, so the pathway really just kind of tells you that this is a multi-step process. Okay, so it can be get quite complicated. But in general, you have 3 steps, right, in the transduction pathway. First one is binding, ligand receptor binding. Okay, so I'm not going to write all of it, so I'm just going to say binding. You got to have that. Okay? And then the second process, it can be very complicated, but we're just going to say that's the signaling transduction pathway. Okay, so that's the transduction pathway. And then the last step is it's going to trigger, that's the end result, right? The end outcome. So that's going to induce a cellular response. Okay, so that's the 3 general steps. Ligand receptor binding, and that's going to activate the transduction pathway, and then last you have a cellular response.